Welcome to this video on visual cycle. The visual cycle is the pathway in which all transretinal produced during phototransduction is converted into 11-cis retinal. Since this was discovered by George Wald, who won the Nobel Prize in Medicine in 1967 for this work, this is also called as Wald cycle. In this topic, I will discuss the biochemical reactions involved, where they occur in the body, and why they matter. Just to introduce the topic, you noticed in my previous video on phototransduction that when light falls on rhodopsin, it converts 11-cis retinal into all transretinal in rhodopsin. And this rhodopsin now called meta-rhodopsin 2. From meta-rhodopsin 2, all transretinal detaches, leaving behind opsin protein part, and therefore all transretinal must be converted back into 11-cis retinal through a number of biochemical reactions which together form a cycle known as visual cycle or Wald cycle. 11-cis retinal then combines with opsin, forming rhodopsin to set in the reaction. I will identify the enzymes carrying out these reactions, their cofactor and locations in the next slide. Just a brief introduction to visual cycle. There are total 4 reactions and there are 2 locations where they occur. Reaction number first occurs in photoreceptor cells such as rod and cone cells, and the remaining reactions occur in retinal pigment epithelium. So when light falls on rhodopsin, 11-cis retinal is converted into all transretinal. This all transretinal in photoreceptor cells converted into all transretinol. This is a reduction reaction, and this is catalyzed by alcoholic dehydrogenase or retinol dehydrogenase. The enzyme utilizes cofactor NADH. All transretinol is converted into all transretinyl ester utilizing enzyme lecithin retinol acyl transferase. Now this all transretinyl ester is converted into 11-cis retinol using an isomerase called RPE65, retinal pigment epithelium 65 enzyme. This enzyme is clinically very important, interesting, because mutations in the gene encoding this enzyme have been identified to cause a congenital blindness known as Leber's congenital amaurosis. This 11-cis retinol is then finally oxidized to 11-cis retinal utilizing alcoholic dehydrogenase or retinal dehydrogenase. In this case, the enzyme utilizes cofactor known as NAD+. 11-cis retinal is photoreceptor, or you can say it receives the light and initiate the reaction. So this can combine with opsin, forming rhodopsin to reset the reaction. So this is all about visual cycle, 4 reactions and total 4 reaction and 2 locations. The first reaction happens in photoreceptor cells, rod and cone, and the remaining 3 reactions happens in retinal pigment epithelium. The visual cycle is highly significant because it regenerates 11-cis retinal, which sustains phototransduction and light vision. Therefore, defects in the enzyme are likely to disrupt this cycle and phototransduction, causing blindness. For example, genetic mutations in RPE65 cause Leber congenital amaurosis, which is kind of complete blindness. Therefore, to realize full significance, the cycle must be learned in conjunction with the phototransduction. Thank you very much for watching this video. I request you to like it, share it, and subscribe it. Until next time, you take care. Bye bye.